So, I guess without further ado, allow me to introduce our first speaker. It's none other than our very own Dr. Byrappa Venkatesh. He obtained his PhD degree from the National University of Singapore. He pursued his postdoctoral studies under the supervision of Nobel laureate Dr. Sidi Brenner and at the Medical Research Council, Cambridge in United Kingdom, where he actually started working on the puffer fish genome. He then returned to Singapore in 1992 and continued working on the pufferfish genome at the Institute of Molecular and Cell Biology at A*STAR. So Dr. Venki right now is currently a professor and research director in IMCB, as well as an adjunct professor of the Department of Pediatrics at National University of Singapore. In addition, he is also the chairperson of the Genome 10K project, which aims to catalog genomes of 10,000 vertebrates. His current research focuses on using a comparative genomics approach for better understanding the structure, function, and evolution of the human genome and the genetic basis of phenotypic diversity in fishes. So without further ado, ladies and gentlemen, let us welcome Dr. Venki, please.
Good evening. First, I should thank the organizers for giving me this opportunity to speak at this unique meeting that's being organized in Singapore for the first time. And I hope this tradition will be continued over a period of time and make this a part of Singapore's science scene. And second, I would like to thank the moderator for introduction. So we can't ask for a better venue than this to talk about evolution of fish. So those of you who have been following this lecture series might recall that the previous speakers spoke up to the evolution of jawed vertebrates, starting from jawless vertebrates to jawed vertebrates. And the next major group is fishes. Fishes come under the category of Osteichthyes, which literally means bony fishes. So as you can see, we are also grouped under this taxa. So that makes us all one type of bony fish. In any case, our evolutionary history is rooted in fishes. In fact, some of the fishes are more close to us than to other fishes, as you can see in this first branch. So in order to understand the evolution of humans, we need to first study the evolution of fishes. Fishes as a group started originating in the Devonian. It's about 420 to 360 million years ago. And number of fishes originated in this period and then further diversified. So this era is known as the age of fishes. And then we had many other branches that took off in subsequent era. So mainly we have two branches in fishes, the Sarcopterygians, or lobe-fin fishes, which led to the land animals, and Actinopterygians, or ray-fin fishes, which led to the large group of living fishes. First, I'll spend some time on the Sarcopterygian, as it's an important branch for us because we are the product of that branch. If you look at this, there are only two fishes represented on this branch. So these two are called the lobe-fin fishes. One is a coelacanth, other one is a lungfish. They're known as lobe-fin fishes because they have a fleshy stem-like base in the fins. It's not very obvious in this slide. So that's why they are known as lobe-fin fishes. So, which is the closest fish to us? This is a major question in evolutionary history, and work has been done on over 3 decades to determine which is the closest fish to humans. So if you follow the evolution of this group, at some point in time, one of their relatives decided to walk out of water. So probably to avoid nagging by its partner, or probably for because there was plenty of food on land. There was very little competition for food and space, and virtually no predators. So this was a major transition point in evolution of vertebrates. It enabled vertebrates to colonize the land. So which is the closest fish to us? There are 3 possibilities. One, coelacanths is the closest relative of humans and other land animals. Second, lungfishes are the closest. Third, coelacanth and lungfish, they form a monophyletic group, and they both are at equal distance to us. So these are the 3 possibilities. So all the work done so far are mainly based on morphological characteristics or paleontological evidence. And to some extent, some molecular data was used to address this issue. Now, in the age of genomics, we are in a position to generate a large number of protein and nucleotide sequences, which have the potential to resolve this kind of complex relationship. So it was decided to sequence one of these fishes and to get a large set of data set so that we can use it for addressing this main issue. Here, the obvious choice was coelacanth, because as you can see, the coelacanth genome is about 2.9 gigabases, which is almost similar to the size of the human genome. But if you look at the lungfish, it has an humongous genome ranging from 40 gigabase to 130 gigabases, which is like 10 to 15 times the size of the human genome. So it's not clear why a lungfish, which is only this size fish, which lives in swamps, freshwater swamps, requires so much DNA in its cell. In fact, all other vertebrates, including us, the so-called highly evolved species, we can manage with 3 gigabase of DNA, whereas these fishes have accumulated about 40 to 130 gigabases. So this is a major puzzle in evolutionary genomics. Probably we'll know the answer when the lungfish genome is sequenced. So what we and others, it's a part of an international consortium, did was to sequence the genome of the coelacanth and then generate a lot of RNA-seq transcripts from lungfish by extracting RNA from different tissues of the lungfish. So here, the best part of lungfish is relatively easy to get lungfish. They're found in all the southern continents, in Australia, Africa, and South America, whereas it's extremely difficult to get coelacanth. But still, we managed through our international collaboration. And in 2013, we were able to generate a good assembly of the coelacanth genome. Coelacanths were thought to have become extinct for a long time because they were not represented in fossils for the last 70 million years. They were abundant around 300 million years, plenty around 200 million years, then they just disappeared from the fossil record. So it was thought to be extinct. Then all of a sudden, in 1938, they found a live specimen in East London of South Africa, and that was one of the major discoveries, or highly sensational discoveries in zoology. And then subsequently, many individuals have been found in South Africa, and more recently, another species of coelacanth has been found in Indonesia. So we know at least there are 2 species of coelacanth, and it is living. So it's generally known as a living fossil. That's because the present-day coelacanth looks very much like its extinct relatives that lived more than 200 million years ago. It has gone through very little change. So it's sort of static in terms of its morphology. So after sequencing the coelacanth genome, we had access to a large number of protein coding sequences. And we used that together with the sequences we had from lungfish to determine the phylogenetic relationship of the coelacanth, lungfish, and the land animals. And this is a tree. My apologies if this looks a bit complicated to some of you. Anyway, it's like a tree, so just follow this. So this tree, based on about 250 genes, showed that lungfish is the closest living fish to land animals and not the coelacanth. So this makes lungfish a very important model for understanding the origin of land animals. But as I said, the genome is so large, we have to wait for some time before we can sequence the genome and compare it with land animals and other fishes and find out what happened in these intermediate stages. So this transition from water to land was a major event in the evolution of vertebrates that enabled the animals to colonize the terrestrial habitat. So this transition was accompanied by a lot of changes in the morphology, anatomy, and physiology of fishes, like fin to limb transition, aquatic breathing to aerial breathing, escaping dehydration, or even managing nitrogenous waste. In water, fishes convert nitrogenous waste into ammonia, which is highly toxic, but it gets easily diffused into the water and gets diluted and distributed in the environment. So this was a major challenge for land animals to excrete nitrogenous waste. So they developed a way to convert this into urea or uric acid, which is less toxic, and developed a mechanism to excrete them on land. So like this, a lot of genetic changes have occurred in line with the morphological changes and physiological changes. So we asked the question whether we can see signatures of any of these changes in coelacanth. So we did find a couple of signatures, and I'm going to share one with you. So one of the transitions that occurred was from fin to limb. So this enabled land animals to raise themselves above the ground and move around freely on the land and explore the terrestrial environment. So as you can see, land animals' limbs are made up of major 3 parts: the stylopod, zygopod, and autopod. Autopod is the one which includes the wrist and fingers, digits. So this is the part that is absent in fishes. Fish fins do not have digits, they don't have wrists. So this is the part that is missing. So development of this autopod, wrist and digit, is actually regulated by a group of genes called Hox genes. There are 4 clusters of Hox genes. These are all developmental genes. And out of that, the Hox-D genes, the cluster of Hox-D genes, regulate the development of autopods. The cluster of genes here, so they drive the developmental patterns of digits and wrist. So these genes are expressed in 2 phases. We call it as early phase expression and late phase expression. The early phase expression drives patterning of the stylopod and the zygopod, whereas the late phase expression is involved in development of digits. So this expression pattern is actually driven by enhancers or regulatory elements. They are located far away from these Hox genes. The genes are here, and the regulatory elements are located in this large region. It's known as the gene desert. In fact, there are 4 clusters of enhancers. They are known as island 1, 2, 3, and 4. So these enhancers drive expression to the different parts of the limb, in particular the autopod and the putative wrist. So we can actually visualize these islands as conserved non-coding elements. The screen is a bit small, so you may not be able to see this. What we have done is we align this region from coelacanth, human and other land animals, and several fishes. And we found that we can find these islands as conserved elements. The pink peaks you see here, they're all those conserved elements. So 2 of them are found in all animals, all the way from fish to humans. Another 2 are found only in land animals. The very first one, if you see, it has a small pink peak in coelacanth and land animals, but nothing in fishes. So this element seems to have evolved in coelacanth and then expanded further to drive some sort of expression. So what we did was we extracted this element from the coelacanth genome and linked it to a reporter gene and expressed in a transgenic mouse. And lo and behold, this was able to drive expression in a limb-specific manner to the autopod region. So this is very significant, considering that coelacanth does not have autopod, it does not have wrist and digits, yet it has this regulatory element in its genome. So what seems to have happened is during evolution, this enhancer, which was present in the coelacanth genome, was recruited by land animals, more elements were added on, and they developed a regulatory network for giving rise to the autopod. So in this respect, coelacanth is an intermediary at the molecular level between aquatic species and terrestrial animals. Now I will move on to the other branch of the tree, the actinopterygians, the ray fin fishes. So while the sarcopterygians were exploring the terrestrial environment, the ray fin fishes started diversifying and dominating the aquatic habitat, and they gave rise to a major group of fishes called teleos fishes. They are this big group here. So one way to define what teleos fishes are is to exclude these 3 groups here, the polypteriforms, acipenseriforms, and holosteae. So if you leave out these 3 groups, whatever other fishes you can think of or whatever you're seeing here, they are all teleos. So this is a way to define it. So this is one of the major groups of vertebrates, and these fishes started evolving in late Triassic and then Jurassic. The major expansion occurred during this phase, and the expansion was so successful, it gave rise to the largest group of vertebrates. So in fact, these 3 groups together, they have less than 50 living species as of now. Whereas these teleos fishes alone, there are about 30,500 species. So that's the largest group of vertebrates. And if you compare it the level of monophyletic groups, you'll see that teleos fishes account for nearly half of all living vertebrates. The land animals altogether constitute another 50%, and low fin fishes and non-teleos another 1 or 2%. So this large group of fishes are also the most diverse group of fishes. You have to just look at the aquarium behind me. They show remarkable variation in their morphology, coloration, in their size, behavior, habitat, and so on. And also these fishes are known for their adaptive radiation. So adaptive radiation means a single lineage diversifies to give rise to several ecologically varied species that can exploit different ecological niche in an environment in a relatively short period of time. The teleost fishes are known for this kind of adaptive radiation. So what is the genetic basis of all this speciation and adaptive radiation? How did teleost diversify into so many species? Of course, this is a very major question in evolution, and we have tried to answer it in a small way, and I will show you some of the data we have generated over the last few years. So one major difference between these fishes, the teleost and all other fishes, is this lineage has undergone a whole genome duplication. That means the entire genome was duplicated once, and they all had two sets of all the genes. So this happened about 300 to 320 million years ago. And as a result, all these fishes have two copies of many genes. So often it has been said that this whole genome duplication is responsible for the speciation and diversity of teleost fishes. There are some examples, but still we don't have very conclusive evidence to link this whole genome duplication to the speciation of teleost. In fact, there is much more stronger evidence in case of flowering plants, and to some extent, actually the origin of vertebrates themselves. At the origin of vertebrates, there were two rounds of whole genome duplication that gave rise to the whole complex system of vertebrates. So if not whole genome duplication, what others could be contributing? So before that, I will show you some evidence which suggests that whole genome duplication may not be responsible for the diversity of these fishes. The first evidence is based on comparing phenotypic variation between holosteans and teleost. Holosteans are the sister group of teleost, the group just before the teleost. And as of now, there are only 8 living species, and they do not exhibit a great variation in the phenotype. But if you consider all the fossil holosteans and compare them with the existing teleost and the fossil members, then you can see that phenotypic variation of holosteans is almost comparable to that of stem group teleost, as well as the crown group teleost. So this suggests that this whole genome duplication was not responsible for the great diversity of phenotypes in the teleost. Second, if you look at how these teleost species have diversified, the rate of diversification has not been very uniform. In fact, the major expansion has occurred just in 2 groups of fishes. One is Asteriophyceae at the top, other one is Pacomorpha. So these 2 groups together account for more than 80% of the teleost fishes alive today. So this suggests probably the whole genome duplication, which occurred much at the base of the teleost, is not responsible for these 2 major diversification. Furthermore, as you can see, there is a time lag between the whole genome duplication and these 2 major diversification. The first diversification of Asteriophyceae occurred about more than 100 million years after the whole genome duplication, and the second one occurred about 100 million years from the present. So there's a big time lag between the time of whole genome duplication and these major diversifications, which again suggests that the whole genome duplication was not responsible for this diversification. And third, we always cite cichlid fishes. Cichlid fishes are one of the most diverse group of freshwater fishes, and there was no whole genome duplication in the recent ancestor of these cichlid fishes. So there are more than 2,000 species of cichlid fishes. They evolved during the Gondwana land, and then when the land migrated and fragmented, they got distributed on, say, India, Madagascar, Africa, Central and South America. So these fishes among themselves, they exhibit great variation in coloration, feeding habit, lip morphology, body shape, and so on. And also they have a very fascinating aspect to the evolution of these fishes. They exhibit what is called evolutionary parallelism. That means even though these species are evolving in different environments, for example here, these species come from Lake Tanganyika, and these species are from Lake Malawi. But if you look at the coloration and body shape, they look very similar. So there is a great convergence of forms in these independently evolving teleost. That has happened repeatedly in many environments. In fact, if you look at the lower pictures here, this thick-lipped phenotype is found in cichlids in Lake Victoria, Malawi, Tanganyika, as well as in Nicaragua. It is all the way in Central America. So this is a very fascinating aspect of evolution of cichlids. How did they evolve similar forms in spite of them being evolving independently? In fact, cichlids are textbook examples of adaptive radiation and explosive speciation. And this has occurred mainly in the lakes, Great Lakes in East African cichlids. In East Africa, there are three major lakes, Lake Victoria, Lake Tanganyika, and Lake Malawi. Out of these, Lake Victoria is the youngest one. And in this lake alone, there are more than 500 species of cichlids. They all evolved within the last 15 to 100,000 years, which is the fastest rate of speciation you observed in any vertebrate lineages. And Lake Malawi, Lake Tanganyika has another 250 species. Lake Malawi has more than 500 species, which also evolved during the last 5 million years. This is again another highest rate of speciation that you can observe in any vertebrate lineages. So these species have gone through repeated cycles of colonizing a new lake, expanding in population, fragmentation, condensation, and again when the lake dries up, they all shrink to a small population, and when the fresh water comes in, they again expand. So it has gone on repeatedly, and whenever there's a new environment, ecological niche created, these people are able to take advantage of that and develop new ecological types that can make use of the food items present in these environments. So again, the question here is, what is the genetic basis of the diversification of cichlids? So what is special about cichlid fishes? What drove them to differentiate into so many species in such a short period of time? So to answer this question, Cichlid Genome Project was started, and we selected 5 species, one each from 3 lakes, Lake Victoria, Tanganyika, and Malawi, 2 from riverine environment, one from River Nile, and the other one was closer to Lake Tanganyika. And genomes of these 5 cichlids were sequenced, and they were compared among themselves as well as among all other fishes to see if we see any signatures that can hint why these fishes are diversifying so rapidly. So I'll just summarize the results instead of going into the details. So first thing we found, the protein coding sequences in these fishes are evolving faster. That means they're accumulating changes faster, giving rise to new amino acids. Obviously, some of them were beneficial, were retained, and used for evolving new phenotypes and diversifying as new species. Second, we found a lot of insertion of repetitive elements. These transposable elements are found throughout the genome. But in case of cichlids, we found these insertions occurred mainly at the promoter region. Promoter region is one which drives the expression pattern of the gene. So because of the insertion of these elements, the expression pattern of the gene was altered. So this has the potential to give rise to expression in a new domain and possibly into a new function. And third, we found an increased frequency of gene duplication in the cichlid lineages. Gene duplication occurs all the time in all the lineages. And here we are talking about tandem duplication, not whole genome duplication. So in cichlid lineages, we specifically found there was an excess of tandemly duplicated genes. And more interestingly, more than 20% of the newly duplicated genes exhibited a new expression pattern. That means after a new copy was evolved, it acquired a new expression pattern and started expressing in a new tissue or a new domain, giving rise to a new function and probably a new phenotype. And finally, regulatory sequence evolution. We found the regulatory regions of these fishes are also evolving faster. So this has a high potential for giving rise to new phenotypes, because this will not affect the protein function, but it will only affect where they are turned on and turned off. So without affecting the main function of the target gene, it can give rise to new function. So I'll show one example of this fast evolving regulatory element. My apologies if you can't see the sequences. What we have done is we took an enhancer from these 5 fishes aligned with Medaka, which is a non-cichlid. And we found that this sequence is highly conserved in Medaka and 4 other species of cichlid, except in one of them, where you can see a lot of blue spots. So it had accumulated a lot of substitutions in this particular species. And then we tested this element. We took one from the Nile tilapia, which is highly conserved, and the one which is very divergent in this P. nairere species, and tested them in transgenic zebrafish. And we found that the divergent enhancer sequence was functional, and it was actually driving expression to a new domain, in this case in the foreplate. The original enhancer was driving expression in the optic tectum and whereas the new one was driving expression in the foreplate and in the heart. So probably it had a new function in these two new tissues. So this way, the genome of cichlids seemed to be somewhat plastic. It had accumulated a lot of variation to start with because of relaxed selection. And there's also some evidence to suggest there were hybridization events very early in the evolution of these species. So when you hybridize two species, it brings a lot of genetic variation. And these fishes were able to exploit that. Overall, we can summarize the factors behind the speciation into three. Unfortunately, the first one is not visible. This is the plastic genome. Second one is the phenotype, especially in terms of sexual dimorphism. Cichlids are known for male nuptial colors and elaborate nest building habits. Also, the males exhibit very rapid evolution in mate selection. So this sort of drives a reproductive isolation between populations and promotes formation of new species. And third, most important, is ecotype. There should be ecological opportunities. When a new ecology is created, a new environment is created, these fishes are all set to exploit that and give rise to many ecological variants which exploit the different trophic levels in this new environment. Now I'll move on to another strategy we have been using to understand the genetic basis of phenotypic variation in fishes. That is by sequencing extreme phenotypes. So by extreme phenotypes, I mean fishes that exhibit very extreme phenotypes compared to all other fishes. A typical example is the family Syngnathidae, which includes seahorses, pipefish, and sea dragon. So this is the tree of the entire family. There are about 300 species. The whole family exhibit very highly derived features. One common feature of all these fishes is male pregnancy, which I will talk about a bit later. So if you look at the morphological phenotype, they exhibit a continuum of variation from one species to another species, reaching its maximum specialization in seahorse. And seahorses, as you all know, and I'm sure you observed on the way here, look very different from other fishes. So this is, if I compare with a normal fish, this is how the comparison goes. Of course, first and most obvious difference is its vertical posture. All other fishes swim horizontal, and this swims in a vertical posture. Second, seahorses have no teeth. Instead, the two jaws have fused together to form a tube-like mouth. So they use this like a straw to suck small food items like zooplankton. Then they have no scales. Instead of scales, the entire body is covered with bony plates all the way from jaws to the tip of the tail in forms of rings that gives them protection, more protection than any other fish can get. And then they have no pelvic fin. Fishes have two paired fins, the pectoral fin equivalent to our arms and pelvic fin equivalent to our legs. So seahorse does not have pelvic fin. It has lost both its pelvic fin. It has also lost its caudal fin. In other words, the tail fin. It has no tail fin like other fishes. And also it has a prehensile tail, which uses it to hang on to seaweeds and corals and stay static in a particular environment. And finally, the males carry a brood pouch on its ventral surface. So seahorses have a very unique breeding process. The males and females mate together, and here there's a reversal of roles here. Here the female transfers the eggs into the male brood pouch, and then onwards, the male looks after the brood until they are fully grown. They are nurtured inside the brood pouch. Brood pouch is functionally equivalent to uterus in mammals. So the brood pouch provides them with oxygen, helps to get rid of nitrogenous waste, and maintains the osmotic level in the brood pouch. So at the end of the term, the males go through parturition process and give birth to young seahorses. And one male can give birth to anywhere from 20 to 1,000 young ones in one pregnancy. So such a large number of young ones can be looked after by these males. So all this makes seahorse a highly derived, the extreme phenotype among fishes. So we thought by sequencing the seahorse genome and comparing with other fishes, we may be able to learn something about how these derived features evolved. So we sequenced the genome of a Singapore seahorse. It's called the tiger-tailed seahorse, Hippocampus comes. It's very common in Singapore, of course, as well as in some Southeast Asian countries. This is one of the species listed as vulnerable. It is sort of endangered because there's a big demand for the seahorses in traditional Chinese medicine. So it's time we generate the sequence of this and see whether what we can learn about the population and whether we can come out with conservation measure to maintain this population. So we sequenced this genome of the tiger-tailed seahorse and compared with all other fishes. So first thing we did was to look at the evolutionary rate of protein sequences in seahorse. So if you can see all these values on the tree branches are paired distances with an outgroup, the spotted gar, and seahorse has the longest branch. Of course, the numbers are very small, but they're all statistically significant. So in summary, the seahorse protein sequences are evolving at a faster rate, something like the cichlid fishes. In fact, seahorse proteins are evolving faster than cichlid. The second fish in the list is Nile tilapia. So seahorse proteins are evolving faster than even cichlid proteins. Likewise, we found even the nucleotide sequence is evolving faster in seahorses. Protein sequence is the one which make the protein, whereas the nucleotide sequences make up the regulatory regions and other regions of the genome. So we found even the nucleotide sequences outside the protein sequences are evolving faster. So what this suggests is there is a high turnover of nucleotides in the genome of seahorse. So there's a high turnover, and probably it's using this turnover to give rise to new elements, new functional elements, and use them for developing this highly derived phenotype. Then we looked at this particular family of genes called secretory calcium binding phosphoproteins, SCPP in short. These genes are important because they're involved in skeletal mineralization. They're important for formation of bones in our body. So all bony vertebrates, that means all the vertebrates that have bony skeleton, they have this family of genes. The number of genes varies from 2 to 20 or even more. And sharks, which do not have bones, whose endoskeleton is made up largely of cartilage, completely lack this gene family, whereas all the other bony fishes contain few members of this gene family. There are two types of SCPP genes. One is acidic SCPP gene, other one is proline glutamine or PQ-rich SCPP genes. These acidic genes are involved in mineralization of bone and dentin, that's part of our teeth. And PQ-rich genes are involved in enamel formation, that's again crown of our teeth. And in fishes, enamel is in teleos fishes, enamel is replaced by enameloid, which is similar to enamel. So these genes are involved in enamel formation. The human genome has a cluster of about 10 of these genes. They're all found in one cluster on chromosome 4. When we looked at these genes in seahorse and all other fishes, we were surprised to find that seahorse has lost all its PQ-rich genes, the one in green. So all other fishes have 8 to 10 members of this gene family, and seahorse has completely lost these genes, except one, which is actually a pseudogene. We could only see part of the gene left. It's a remnant of a gene. So essentially, it has no PQ-rich gene. So PQ-rich gene, as I said, are important for the formation of enameloid or enamel. So we propose that loss of this gene family has led to the loss of teeth in seahorse. So loss of teeth is not new in vertebrate evolution. This is known as a condition known as edentulism, loss of teeth. For example, all birds have lost teeth. All turtles have no teeth. Some of the mammals like baleen whale and pangolin, they don't have teeth. So when their genomes were sequenced, scientists did find there was mutation in these enamel-related and dentin-related genes. Now we add one more group to this, that is seahorse, which has lost all the genes, and it has also no teeth. So this provides further support to our hypothesis that loss of these genes led to loss of teeth in seahorse. Then loss of pelvic fin. I said seahorse has lost pelvic fin, and pelvic fin is equivalent to our legs, whereas pectoral fins are equivalent to our forelimb. There are 4 transcription factor genes that are important for the formation of forelimb and hind limb. PITX1 and PITX2, these are homeobox transcription factors that are important for the sustained growth of hind limb or pelvic fin, in other words. Whereas TBX4 and TBX5, these are T-box family of transcription factors. TBOX5 is important for development of forelimb, whereas TBOX4 is important for development of hind limb or pelvic fin. So our candidates for the loss of pelvic fin were PITX1, PITX2, and TBX4. We look for these genes in the seahorse genome. And interestingly enough, seahorse has completely lost TBX4 gene. This is a cluster of genes that are highly conserved in all the fishes and, in fact, all the vertebrates. So if you look at this alignment, there's a TBX gene in other fishes, and there's a gap here. That means the gene has been completely lost. So we hypothesize that loss of this transcription factor is responsible for loss of pelvic fin in seahorse. So to test this hypothesis, we knocked out this gene in zebrafish using the CRISPR-Cas9 method, which is a very powerful gene editing method. So we introduced mutations into TBX4 gene in zebrafish. And if you look at the mutant, which has lost both copies of the TBX, it has completely lost the pelvic fin. This is the wild type, which has a pair of pelvic fin, and the mutant has completely lost pelvic fin. So this suggests that loss of TBX4 was responsible for loss of pelvic fin in seahorses. In fact, pelvic fins have been lost several times in evolution. For example, eels, they don't have pelvic fin. Seahorse and pipefish. Pipefish also belongs to the same family. They don't have pelvic fin. Some populations of stickleback, especially freshwater stickleback, they don't have pelvic fin, whereas the marine counterparts have pelvic fin. And all the tetradontiform fishes, like the pufferfish and ocean sunfish and so on, they also lost pelvic fin. So we now know that loss of TBX4 is responsible for loss of pelvic fin in seahorse. But in other fishes, their genomes have been sequenced, and the TBX4 is still intact. Of course, we know previously it has been shown that a mutation in PTX1 regulatory region in stickleback was responsible for loss of pelvic fin. So what this suggests is evolution targets different genes or different network to arrive at the same phenotype. That's in this case the loss of pelvic fin. So there's no single way to arrive at a particular phenotype. And finally, the male pregnancy. So the male brood pouch is a highly specialized organ. So because it has to nurture young ones for a long time until they are fully developed. When this brood pouch develops, the epithelial cells undergo a lot of developmental changes. There'll be proliferation of epithelial cells, development of some secretory cells, and extensive angiogenesis. And these changes are also accompanied by changes in the genetic makeup. There's a lot of things happen in the genome. So we were wondering whether we can see any signatures of these changes in the genome of seahorse. So we looked at many genes, and we came across this family of genes called astaxin metalloprotease subfamily. So these genes are known to be involved in hatching of eggs in other fishes. So they're called as hatching enzymes, high coriolytic enzyme, and so on. In pipefish, somebody had sequenced one gene from this family, and they found it was expressed in the male brood pouch in pipefish. So they gave a new name to that gene called patristacin. Patri means paternal, patristacin. So when we analyzed this gene family in the seahorse, we found that this family has undergone an expansion in seahorse. So right at the bottom, you see these red arrows. Seahorse has about 6 of those genes, whereas all other fishes have 2 or 3 copies of these, except platy fish, which is just above seahorse. I'll talk about that in a minute. So in seahorse, it looks like this gene expanded by tandem duplication. So what does this newly evolved gene do? Are they functional? Where do they express? So when we looked at their expression in various tissues, we found that 5 out of these 6 genes expressed in the male brood pouch, highly specific expression in the male brood pouch. In fact, they show differential expression at different stages of brood pouch. Some express high in the mid-pregnancy, some at late pregnancy, and so on. So these data suggest there's a link between these new genes and the development of brood pouch. So this is one interesting aspect where genes have expanded through tandem duplication and acquired a new expression pattern in a totally new domain, because its ancestor fish had nothing like brood pouch in the males. So it acquired a totally new function and helped in the development of this brood pouch. So what about platy fish? So interestingly, platy fish also has 6 copies of this gene, but the expansion has occurred independently in the 2 lineages. If you look at this tree, seahorse and platy fish are not on the same branch. So that means it has independently evolved. So what is special about platy fish? Platy fish, if most of you know, it's a ovoviviparous fish. So just like seahorse, it has internal fertilization, of course, in the females, and female carries the young ones until they're fully grown and then give birth to young ones. So this is ovoviviparity. We can call it as female pregnancy. So it's interesting that in the evolution, same family of genes have been targeted to develop similar phenotype, that is male pregnancy in seahorse and female pregnancy in platy fish. So this is how evolution uses various genetic tools it has its disposal to give rise to this new phenotype. To summarize, what are the genetic changes we observed in the seahorse? It has a higher molecular evolutionary rate. That means there's a high turnover of nucleotides. It has lost some crucial genes. Some genes have undergone expansion, and also they have acquired new expression pattern. And then there's a high turnover of regulatory elements. I didn't show much data about this, but seahorses have experienced loss of many ancient regulatory elements and gained a lot of new elements. So finally, coming back to the fish. So such a large variety of fishes, if you look at them, it looks like it's a result of nature's experiment. Nature seems to have carried out a random mutagenic experiment, created hundreds of lines of these mutagens, and passed them through selection. And at the end of that, we have all these diverse species. So as Sidney famously said in his Nobel Prize speech, these are nature's gift to science. So it's for us, to scientists, to interrogate these genomes. Now the cost of sequencing has come down so much, you can virtually sequence any genome. It's a matter of little $1,000 more or less, but you can sequence any genome. And with that, comparative genomics becomes a very powerful approach. You can compare with the other species, other fishes, and then use gene editing technology to recreate that in a model species and understand the genetic basis of this variation. So with that, I would like to conclude my talk, and thank you for your attention.